What are the factors that affect diffusion? First is temperature. The higher the temperature, the higher the diffusion rate. So when, for instance, another example, there is this potassium permanganate, the one that this purple colored dye that is used in clothes, or blue color. When you put that dye inside water and you put it down, and you also put it in another water and put it on a stove, you discover that the one that is put on the stove will dissolve faster, and the dye will spread faster in the one that has it added than the one that is just there naturally. So high temperature aids diffusion rate. Then another thing is difference in concentration. Molecules move fastest when concentration differences are greatest. For instance, what concentration difference is? We talked about the high and the low concentration. The high concentration is when a particular substance is so concentrated in a particular place compared to another where that thing is not there at all. So when the difference in the concentration is so high, it will be easily felt than, as in the diffusion rate will be faster than when the concentration is low. For instance, we are passing a particular substance through a plate. When you have something that is, the concentration is medium, when the concentration of a particular substance is medium, the diffusion rate is slower than when there is no concentration at all. That thing will move very fast because it's neutral, unlike when you have that substance you are trying to pass across in a minute concentration. Also, another factor that affects diffusion is the molecular size. The smaller the size of the molecule, the faster the diffusion rate. When you have a small molecule, diffusion occurs faster in that than when the molecule is large. Then another factor is the state of the matter. We have three states of matter. We have the solid, the liquid, and the gas state. So diffusion rate is slowest in solid because solid is compact together. While in gases, gases are free. They are free. You can just find them anywhere. It's fast in gases, but it's also fastest. Diffusion rate occurs fastest in liquid, followed by gases. Then in the least is in solid. The rate of diffusion in solid is slower compared to gases, but liquid is the fastest. The second exchange process that occurs around a cell is osmosis. We have talked about diffusion. Moving on to osmosis. Osmosis is the physical process by which water solvents move from a dilute solution to a region of higher concentration across a semi-permeable membrane. When water solvents move from a dilute solution into a region of higher concentration. Now, in osmosis involves water concentration. It occurs with water concentration in solution. It occurs in solution, unlike diffusion. Diffusion can occur in liquid, solid, and gas, but osmosis majorly is in solution, and it involves a semi-permeable membrane. The semi-permeable membrane allows the solvent molecule to pass through. So it allows some things to pass through. It's not everything that you allow. Just like a sieve, when you pour a mixture of your gari and water in a sieve, the gari will stay. So that gari is said to be the solvent, while the water is the solvent, while the gari is the solute. So it's the water molecule that moves. And I'm using the sieve as the semi-permeable membrane because not everything will pass through the sieve. So the solvent is the water or the liquid that passes through. So it's the liquid that passes through in osmosis. Then water passage, that is the movement of water, will continue until equilibrium is reached. What is the equilibrium? That is a balance. That is at both edges. Okay, let me use an example. You have salt in a container, then you put sieve, then water is in the other part, such that the water can go through to where you have the salt. Before you know it, the water, when you taste the water will become salty, and the concentration of the salt is reducing. It's not as concentrated as it was when you have not added water. So by the time the exchange is occurring, the water is entering the salt, we get to a state that it will become balanced. The taste of the salt, the original salt and the original water will now become the same. That is when equilibrium is reached, and that is when osmosis stops, because there is nothing to exchange again. In osmosis, we have some terms relating to the cell. So, and the first is hypertonic. When we say hypertonic, what does it mean? Look at this. For instance, this is a cell in a solution. When the external concentration, when this solution, for instance, that the cell is placed, is more concentrated than the contents of this cell, then you say the cell is hypertonic. That is, when the concentration of the solution that surrounds the cell is higher than the concentration of the content of the cell. So we have this being higher. The concentration of this is higher than what we have in the cell. So you say it is an hypotonic. It is an hypertonic solution. When the external solution is higher in concentration than the internal of the cell, you have a hypertonic solution. The next is hypotonic solution. Hypotonic is just the opposite of hypertonic. When the concentration of the cell content is higher than that of the surrounding environment. So you have what we call hypotonic solution. So look at it. The concentration of the cell being higher than the solution that surrounds it, that is when you have hypotonic. But when the surrounding is the one that is higher, the concentration of the surrounding is higher than that of the cell, then you have hypertonic solution. Now, there is also another one we call isotonic solution. Iso means equal, ISO. So when the concentration of the cell and that of the solution is the same, then you have what is called the isotonic solution. That is, the same concentration of the content of the cell is the same as that of the solution that surrounds it. So we have the isotonic solution. Then also we have what we call exosmosis from osmosis. Exosmosis. So you have the EX, osmosis. This is the movement of water molecules from inside the cell to the outer part of the cell. That is X, out. Movement of water molecules. Water is moving out of this cell into the external. That's what we call exosmosis. Then we have endosmosis. Endo, that is internal, which tells you is the opposite of exo. And osmosis, that is when water molecules move from the external, from the outside of the cell into the cell. Water molecules moving from out the external solution or the outer surrounding of the cell into the cell, then you have what is called endosmosis. We will now be considering the examples of osmosis in plants. And the first is seen in the absorption of water from the soil solution into the root hairs. You know, we established that osmosis is the movement of water from a region of higher concentration to a region of from a region of higher concentration, that is where there is no concentration of the other substance, to a region where there is a concentration. So now, example of osmosis in plant is the water absorption. When you put water in the soil, the root hair absorbs the water. So that is an example of osmosis. Then another example is the opening and closing of stomata by guard cells. You know, stomata is the one responsible for respiration in plants, and it is also the one that it controls the rate of water absorption and the rate of dehydration that occurs, transpiration in plants. So when the stomata is opened, there are various factors that affect the opening of the stomata. Light is a major one. Stomata is opened in the day, when there is sunlight, but at night you find it closed. So osmosis is the one that controls that, the opening and the closing of the gas cells of the stomata. Also, the movements of water across the cells of the cortex and that of the leaf is an example of osmosis in plants. Examples of osmosis in animals include the entry of water into the cytoplasm of a single-celled organism like amoeba or paramecium. Water enters, they are aquatic, so when water enters into the organism, into the cytoplasm of that particular organism, that's an example of osmosis. Also, the absorption of water in the large intestine of the animal is also an example of osmosis. When the food is digested and the ones that are not useful goes into the large intestine. In the large intestine, the water is absorbed from the remains of undigested products. So the process of absorbing the water, whereby you now have the solid material going out as fecal material. So that is the absorption of the water is an example of an osmotic example in animal. Also, the reabsorption of water in the kidney tubules is also an example of osmosis. Now, I want to talk about the differences between osmosis and diffusion. I want to look at what makes them different, and also we'll later be looking at the similarities of osmosis and diffusion. The differences between osmosis and diffusion include the following. 1.